Agarose gel electrophoresis. Agarose gel electrophoresis. Separation of molecules based on their charges and size using agarose gel as the separating medium. Agarose. This is the most widely used gel matrix made up of polysaccharide formed with agarose. It is a natural and linear polymer which is extracted from seaweed. It is made up of repeating disaccharide unit known as agarose consisting of 3,6 anhydrogalactose and galactose. The agarose provides a uniform porosity than starch whose low concentrations make large pore sizes while it gives smaller pores in the high concentrations. It may also be differed by modifying the initial concentration of the suspension. When heated with a buffer, it forms a gel matrix on cooling due to hydrogen bonding. Principle: if a direct current is passed through an agarose gel block containing charged molecules, the positively charged cations will migrate towards the cathode and the negatively charged anions will move towards the anode. Proteins and nucleic acids are negatively charged molecules, with the result that they tend to move towards the anode. Mobility of the molecules depend on the net charge, size and shape of the molecule, pH and composition of the suspending medium and electric current. Agarose is a linear polymer of D-galactose and L-galactose. When it is mixed with water, it undergoes cross-linking between the agarose units and forms a porous matrix called gel. If a solution is dispersed within the gel, the pores have sieving capacity. As a result, smaller molecules move towards the anode faster than the larger ones. The pore size is determined by the concentration of the agarose. DNA and mRNA are negatively charged molecules, so the electric field helps them to move towards the anodic end. Procedure: agarose is dissolved and heated by keeping it in a microwave oven. It is then transferred into the trough of electrophoresis apparatus up to the height of 75 mm thickness. Prior to solidification, a comb is fixed at one end to prepare loading wells. The comb is removed to have loading wells in the gel. Buffer reservoirs are filled with 1x TAE buffer. The DNA sample is digested with the restriction enzyme. The DNA sample is heated to 65 degrees Celsius for minutes to remove aggregates into individual fragments of DNA. To observe the progress of electrophoresis, a dye bromophenol blue of known migration rate is mixed with the DNA sample. The sample is filled in the wells and suitable molecular markers are loaded in one well. The electrophoresis unit is connected with the power supply and is switched on to start the movement of DNA fragments from the cathodic end to anodic end. After about 1 hour, the current is switched off and the gel is transferred to a container containing ethylium bromide. The stained gel is washed with distilled water to remove unbound ethylium bromide from the gel. This gel is exposed to UV light by keeping it under an UV transilluminator to absorb light bands that indicate DNA bands. The size of the DNA fragments can be estimated by comparing their locations in relation to the positions of the molecular markers. Applications. It is used for the separation of DNA fragments in a sample. It is used to detect the purity of DNA fragments. It is used to determine the size of DNA fragments. It is also used in the separation of proteins in a solution. Estimation of the size of DNA molecules. Analysis of PCR products, for example, in molecular genetic diagnosis or genetic fingerprinting. Separation of restricted genomic DNA prior to southern analysis or of RNA prior to northern analysis. Agarose cell allow purification of DNA fragments. Thank you.